So today we're going to do a quick biochem review. We're going to talk about the importance of directionality in macromolecules. We're going to have some multiple choice questions at the end, and hopefully time for some free response practice. It's really hard to get a really good free response question in because on the AP exam, it is involved. A lot of the FRQs involve multiple units and content from a bunch of different units. And so since we're in our first unit or two, depending on what class you're in and where, what your teacher's doing, we can really only pull from stuff that we know of biochem, but we're going to still get some practice in if we can, and then we'll have open questions. So real quick, last stream, if you logged in, was just on properties of biological molecules. So our streamer just talked about structure and function and just really focused on what are the parts of a nucleic acid, what are the parts of a protein, how do they go together, same thing with carbs and lipids, and just kind of surface level talked about everything. Today we're going to get into the nitty gritty, and these are the three things I hope you can do by the end of today. So A, describe the directionality of DNA in proteins. B, explain the significance of the specific order of amino acids for proper functioning. So IDA, that's where we're going to really hit the protein structure. And then C, explain the significance of the specific order of nucleotides for the proper functioning of DNA. So every stream ideally builds on the last. And but if there's anything that we're not hitting in the streams, feel free to mention it in the chat, and we can always make a stream topic and run it sometime later in the week. Based on what you guys are wanting. Can I post the link? Yeah, if the PowerPoint slides should be linked already. If they're not, give me 1 second and I can get that linked to you guys right now. Hold on 1 second. Oop, give me 1 second so I can try to share this link to you. Okay, here we go. Is that working? All right, so let me know if that worked for everybody, and I will get us back to where we need to be. That works? Okay, cool. All right, so brief review from last time. This is a big table. However, I think it highlights the most important things that you probably should have gotten from the last two streams. So we went over the 4 macromolecules. So these are the molecules in biology that are the most important. They're not necessarily the only macromolecules, but they're the 4 that we focus on. So we've got carbs, lipids, nucleic acids, and proteins. And the table just outlines here what's the monomer. And so we say mono meaning one. So it's the building block to that larger macromolecule. So by now, hopefully, you should feel really comfortable describing what is the monomer that makes up those 4 macromolecules. And not just the name of them, but potentially even their chemical structure. So if I were to give you a structure of amino acid compared to a nucleotide, in the chemical formula, you should be able to say which one is which one's going to build protein and which one's going to build DNA. Okay. I like having biology examples because a lot of the times in your AP Bio FRQs or free response questions, so I apologize, we call them FRQ, free response question. A lot of times you have to use biology examples. So if you're going to be talking about the importance of the sequence of amino acids that make proteins, you can't just keep calling everything a protein. You have to show that you know your biology and explain, where does this happen in real life? Where is this happening in biology? So if I'm talking about maybe a protein that misfolded somehow, link in something like enzymes, or link in something like antibodies and how that shape is so important. And then also obviously talk about the function of that example. So if you don't have this now, you do have access to the slides. So write it in your notebook, come up with your own table. They're really, really good for reviewing old material. Okay. All right. So I'm just going to briefly review that structure of each macromolecule so you can see what I'm talking about when I say if you see a drawing, you should be able to tell me what I'm looking at. So for example, nucleic acids are made up of the monomers nucleotides. So if you're looking at the slide right now, there are 3 main components to the monomer. So a monomer doesn't just mean it's this one thing that's identical all the way around. Monomers are a building block that builds your polymer, but the monomer itself can have several different components that all have different chemical and structural integrities. So for example, if I were to say which component here is the phosphate group, take a minute and look at that picture. Would you be able to, on a test, like circle what is the phosphate group of a nucleic acid? So if you want, I know you can't circle it and show me what you're doing, but maybe just type it in the sidebar. How would you describe which portion of that nucleic acid or that nucleotide is the phosphate group? So go ahead, let me know, what is the phosphate group of this nucleic acid, of this nucleotide? Awesome, Sen. Yeah, so Sen said, yep, it's a left part, left cross with a P in the center. Good. Yeah, everyone's saying it's that left component. The reason that's going to be important is that if you start talking about the structure of RNA and DNA, like I've pictured here, you need to know which part of the nucleotide is forming that structure, right? So we've got like, what is this ribbon-like material on the outside of DNA? And then what are these colored bars on the inside? What part of the monomer are actually contributing to that shape? And if you're able to talk about that and draw it, and make connections and know what you're talking about, you're going to be way better off. So yeah, if you were to circle that, for those of you who did not get a chance to answer, this is our phosphate group right here. So there's a P for phosphate right in the middle. Remember too, with your macromolecules, be aware of what elements are in each macromolecule. So if we were to say, in my last stream, I talked about an experiment where they identified that there was phosphorus in a test tube, and then the other ones all had just carbon, oxygen, and hydrogen, and you're trying to figure out and then defend which one has the nucleic acid. The only macromolecule that we're looking at with a phosphate group is going to be your nucleic acid. So that's another important distinguish, is not just knowing the structure overall, but then individually, what elements are there, what elements are not there. Okay. Second, what about the nitrogenous base? You don't have to type it in, but just in your head, maybe think about where you would put a box around to label the nitrogenous base of our monomer. So we're looking at a nucleic acid, and we want to know which one is the base. Okay, so hopefully in your head you've thought about it. And right top, yeah, very good, very good. Oops, sorry, my thing is backwards here. What I've got boxed in white is going to be our base right here, right? So we have 4 different bases. If you haven't gotten to that yet, you have adenine, tyrosine, guanine. What did I say so far? A, C, and cytosine. So you have A, C, G, and T. 2 of them are double-ringed bases, and then the other 2 are single-ring bases. And you'll get way more in detail with that with me on our Tuesday stream, because it'll just be about nucleic acids, and we'll really, really break it down. Okay, so we've got the phosphate group circled in teal. The box with the white is our nitrogenous base. Think about why it's called a nitrogenous base. Look at all those nitrogens in there. That's another good one to think about. If you just think of them as bases, that structural component won't really jump out at you. But if you really think back to, oh, it's a nitrogenous base. Okay, if it's nitrogenous, there's probably some nitrogens going on. So look at that. There's tons of nitrogens in there, and that can also help pinpoint you. And then also we have our deoxyribose sugar here. So this is a DNA nucleotide, deoxyribose, looking different than ribose. And we'll get more into that in the next one as well. Okay. Back to just protein is what we had mentioned earlier. The amino acid is our monomer. It has 3 sections as well, or we like to call it 3 sections. If you can, think about which is the amine group and which would be the carboxyl group. So if you haven't done functional groups in your AP Bio class, that'd be a good thing to look up on your own and just look at the functional groups. Yeah, Ida, good job. Yeah, so Ida, thank you for participating. Yeah, the amine group is going to be your NH3. And then on the right side, yep, good, that's the amino group. So there's our amino group. Here's our carboxyl group. And so if you think about what having directionality with a protein, one side of your amino acid is going to be more positive and the other will be negative. Good. So here's our carboxyl group. And so if you think about having two of these next to each other, like I have pictured here, this is where that covalent peptide bond is going to be formed. Okay, so you've got your hydroxyl group and your hydrogen from the amine group. Do you guys remember what that process is called? When water is being taken out so that a new bond can form? Anybody remember what that's called? Yeah, awesome. So it's dehydration synthesis. So this picture is nice. It's showing that we've got a hydroxyl group and a hydrogen. They're going to be taken out. So we're dehydrating the two amino acids, and then there's that peptide bond right there. So now your carbon and nitrogen come together. Okay, and so we would call this left side the positive terminus of the protein, growing protein, and this would be our negative terminus of the other protein. So we'll get more into that later too, but the directionality where you start to actually build your protein, that's a good thing to remember, that it has directionality as well. All right, so now we have lipids. Okay, this circled component, go ahead and type in, what is this component called of a lipid? So lipids are made up of fatty acids and glycerol. So this circled component right here, are we working with a fatty acid or are we working with glycerol? So glycerol is the right answer. Yes, okay, good, Tanea. So glycerol, absolutely. The other component of your long hydrocarbon chains that make up lipids are your fatty acids. And so that's this next portion where you get this long hydrocarbon chain with a carboxyl group at the end. Similar thing will happen, dehydration synthesis. And notice there's 3 hydroxyl groups on a glycerol that can be attached with a fatty acid. And so normally we have triglycerides where we get 3 fatty acids connected to our glycerol, or in the case of phospholipids, so there's my fatty acid, there's my box showing dehydration synthesis where the bonds would form. Oh, I don't have that picture, sorry. We'll get to phospholipids later, but when we do, phospholipids have 2 fatty acid tails, and then one, that other hydroxyl group has been joined in with a phosphate group, a modified phosphate group. Okay, almost done with just our brief review. Here is our monomer to building a polymer for carbohydrate. So in this picture I just showed glucose and fructose coming together, water being taken out, dehydration synthesis, we get this nice covalent bond, and this is a disaccharide. So if we were to keep doing this over and over again and just adding more glucose, or more fructose, we'd get a long chain that could then be used for energy storage. So for example, if you had, like, I've got to be honest, I had a cupcake right before this, right? And so my body probably didn't need all the glucose that was in that cupcake. So once it starts breaking down the glucose, some of it will be used for cellular respiration, so to make ATP, and we'll get into that too in a couple weeks. So some of it is used right away, and a glucose molecule is fabulous for just quick breakdown, making ATP, and then we can do cellular work with that. But to be honest, I'm sitting here right now, I'm not running a marathon, I'm not working out. So with that excess sugar, my blood sugar, which is the level of glucose in my blood right now, it's probably a little high. Doesn't need to be that high. My cells don't need it right now. So what will happen is that my body will send a message to put those individual molecules of glucose together and put it into a form called glycogen. And so glycogen will then be that long-term, short-term storage, depending on how much sugar I've eaten. Obviously, if I have a high sugar diet, I'm gonna have lots of extra glycogen. But it's going to store it in my liver until I need it. And so if I don't eat anything else the rest of the night and I'm hungry at 6 or 7 a.m., but let's say I'm too busy for breakfast and I just head out to work, my body isn't just going to collapse. It's going to be able to break down those polymers of glucose, and so it'll break down glycogen, and glycogen can be used to help my blood sugar feel better. Okay, so if we think about that, let's say I've got this long chain, which I know is not pictured here, but imagine we have a really long chain of these molecules together, and I have a polysaccharide, glycogen. What would be the reversal process that my body needs to do to break those up, to put glucose back into my blood? So putting them together is dehydration synthesis. What's the name of that process that needs to happen to start breaking up the stored carbohydrate? So go ahead and chat that in. Oh, awesome. We have some fast answers. Good. Yeah, so the reversal of dehydration synthesis is going to be hydrolysis, right? We can break that word up into hydro and lysis. Lysis meaning to cut or break. Hydro meaning water, right? So that means a water molecule is coming in here, right over here, and we're going to break that bond. And so we'll end up forming our two individual monomers again. And that is what's happening constantly, right? So constantly macromolecules, especially carbs, can be built and broken. Usually enzyme regulated, so that things aren't just breaking in the presence of water, right? We are mostly water. Our cells live in a very watery environment. So remember not to think of water as this automatic breaker and maker of bonds, because then it would just be chaos and our bodies would be falling apart. So they are enzyme regulated, which obviously will come later in more in-depth live streams. Okay. Also, now let's kind of move from a review to now really thinking about the structure of these things and how that affects the function. So I'm going to start with carbs here because I think they're kind of an easier one to look at and see the connection of like, oh, I see why its structure, which means the way it's shaped or organized, affects its function. And when we say function, we mean its job, right? What is its job in the cell? I like to think of everything that's in a living organism, whether it be a human or a plant or an animal or a bacteria, every little part has a job, right? Even if it's a job to just sit there and do nothing and just be a cell wall and maybe be a little bit fluid and let things in and out, or it can be something totally structural, like I am a part of a tree that helps make the bark and I help withstand the pull of gravity, right? So everything has a job. And so what about its structure allows it to do its job? So, carbs in their polymer form, they can be very linear or they can be branched. They can also be highly branched. So first, if we were to look at cellulose, give me like a thumbs up or like a happy emoji, just so you know what I'm talking about. Would you call cellulose linear? Does this structure look linear to you? Give me like a thumbs up or a high five or something. Show me if you think cellulose from this diagram looks like I did the okay sign. Yeah. So cellulose is linear, right? And so we have these stacks of polymers. And so when they're linear like that, each of those strands has the opportunity to hydrogen bond with the one above or below it. And so it actually causes it to be pretty rigid. Think about what if we go from liquid water to ice, and so remember when we have water moving into ice, those hydrogen bonds kind of stabilize. And so then we get this crystalline, like wider hydrogen bond structure that makes water less dense as a solid. So if we have hydrogen bonds that aren't breaking and moving, and if you think about water is constantly breaking and remaking hydrogen bonds with the water molecules around it. So it's fluid. If we have something that has like pretty rigid cross links of hydrogen bonds, it's going to be really strong. And so cellulose is what is in our plant material for structure. And so it allows plants to grow tall and not fall when gravity is on it, right? Or if you think of even like a strawberry plant and it's got this heavy fruit growing on it now, why does the whole thing not fall over? Because you have cellulose. You have this structural component that, because of its rigidity and its hydrogen bonds and its linear nature, right? It's like very strong pillars. It's going to hold things up. Okay, so that's an example of structure function. It's also something that we cannot digest, right? So cellulose is stuff that we would call like roughage in our diet. So it actually just, we can't digest it. There's bacteria in our intestines that can digest it a little bit, but that's usually what just comes right out. No, I'm not going to go to that detail because that's a gross example. Moving on. Our branched example, we have starch right here. So that's a plant storage example. So plants basically take glucose molecules and they pile them all up together, but then they branch off into these segues, if you will. They're not highly branched like glycogen. So there's not a lot of room for hydrogen bonds between these. So the more you branch off, the more separate these components become. And the polymer itself is not going to be this clump that's hard to break down. The reason we don't want it to be hard to break down is because starch is for energy storage. So if the plant needs that, it needs to be able to access it. And if it's heavily hydrogen bonded and heavily compacted, the plant would have to do a lot of work to break in there and get the energy. So these branches offer, it's almost like on a highway if you're driving and there's all these options, right? So all of a sudden the highway goes from 1 lane to 4 lanes. Traffic moves really nice, right? And it's really easy to access moving lanes and get to the exits and things like that. Same thing with your starches and your carbs. So if we've got these extra highways, the cell has more opportunity to be able to break that down when needed. Okay, glycogen, this is just a human example. It's just a more highly branched example than glycogen, than starch. So this is what I was talking about in my previous example, the glycogen that gets stored in your liver, highly branched, easily accessible, which is really, really good because if someone's blood sugar is dropping, we don't want them just to pass out, right? We've all felt that like crazy hunger and your head kind of starts to hurt and maybe you do get a little dizzy. Even if you don't eat anything, your body will try to raise that blood sugar for you. Right? It's not going to last forever. You have to eat. But glycogen can be broken up into these individual glucose molecules, and then your cells can use those glucose molecules as needed, right into your bloodstream. Your blood will carry it around to whatever cells need it, particularly your brain, right? So if your brain doesn't have any sugar to make ATP, you're going to not feel so great. And so those highly branched, so think again, explain the structure, right? We have a monomer that's built into a polymer, right? So a glucose monomer that's built into a highly branched polymer, right? And so all these branches allow for easy access to break them down when needed. Okay, so that's probably the most in-depth example for carb structure and function. I'll go a little more brief with lipids. Because there's basically just 3 things you need to know about that. We've got saturated and unsaturated lipids. And so if we have something really saturated, it's similar to the idea of like highly branched carbs where it's not a lot of movement. But if it's unsaturated, they're not going to be able to stack together very well. And so you get all these kinks. And so that's where we see, you know, the food example of saturated fat being your like solid fats like butter. And then unsaturated are your things like oils that remain liquid. Okay, and the reason they're liquid is that all these different fatty acids can't clump onto one another and then form hydrogen bonds. They just are always kind of moving around. That keeps them fluid. Okay, so that allows for different things in the body as well in terms of, are we using something for insulation, right? So if we have a lipid in the body or in an organism that's for insulation, you wouldn't want it to be liquid. You would want it to be solid. So those structures probably are more saturated. Saturated versus unsaturated, just in case you were unclear and missed the chemistry of that last time. If you look here in the unsaturated fatty acid, it has a double bond. And so we would say those carbons are not saturated with hydrogens anymore. Saturated means, are they filled with carbons, right? Because we know carbons like to have 4 bonds. So 2 of those bonds are to the neighboring carbons. The other 2 would be to hydrogens. So if we have a double bond, you're not saturated with hydrogens anymore. And then it makes you a little bit more flexible. Okay. The most important lipid right now for the next unit, when we talk about cells, is going to be your phospholipid. And so we've got two, instead of a triglyceride, your glycerol molecule has two fatty acids, right, connected, and then a phosphate group. And so that's going to make something really unique where we have something that likes water at the top and then something that doesn't like water at the bottom. And so when we get into cell membranes and how important that structure and function is, it's quite honestly a miracle that this molecule exists and that every cell membrane has it. Because without it, cells would be leaking and cells would be falling apart. And honestly, cells would have never formed if we didn't have this membrane to protect its internal and external environment. All right, let's keep going. Now we're on to proteins. So for those of you who asked about proteins, here we go. So I actually drew this because, to be honest, drawing your own pictures of what you're learning, everyone, it helps so much. It really does. So even if you think you're not a visual learner, take the time to draw out the stuff that you're reading in your textbooks. Even if it's something your teacher already gave you and it's a worksheet or whatever, redraw stuff and rethink through it, relabel it. And so in those moments of maybe when you're taking a test, you can remember, oh yeah, I remember why I did that dotted bond, or why I represented the bonds with a dotted line rather than just all straight. Like, so for me here, I have 4 amino acids, right? So I have an amine group over here on the left, I have my R group, I have my carboxyl, right? And then I drew a dotted line to remind you all that this is a peptide bond right here. So this is one single amino acid, and then there's a peptide bond to the next amino acid. The dotted line is representing a peptide bond. Not dotted because I need to show that it's weaker or anything like that, because it's a very strong covalent bond, which is why I drew that in here as well. But again, this is how proteins are being built. We have a positive end and a negative end. And I have another picture showing that as well, but they grow from the negative end. And so when you guys get into transcription and translation and the building of actual proteins and how there's enzymes that regulate it and there's certain areas of the cell this is happening, it'll make more sense why we say it grows from one end. It's not just like randomly being put together on the left and the right. It actually grows from one end to the next, and then it starts to fold. And then I just kind of color coded these amino acids because I'm not going to keep drawing the actual amino acid structure to do an entire protein, explain folding. So I like to draw amino acids as just circles. And then we can think about the folding from there. So let's say I just have a simple 4 amino acid. Come on now. Oh, there it is. There's my positive and negative ends. There we go. A positive and negative end right there. So more amino acids would be joined in on the right. All right, so with protein folding, so here's my cartoon example again. I've just got 4 amino acids, super small, but I just didn't want to overwhelm everybody with a bunch of pictures. But if you remember, each of those amino acids has an R group, and that R group has its own structural formula as well. And so there's 20 options for R groups. That's why we have 20 different amino acids. So I just gave an example of what it would look like if we had an acidic and a basic R group near each other. So if you are basic and acidic, you are positively charged and negatively charged. So that amino acid has a little, group hanging off of it that is going to act positively, right? And so if we think about back to even like static interactions, or if you took physics before, what happens when positives and negatives are near each other? Go ahead and type that in, actually. So go ahead and type it in. How would a positive and negative charge behave with one another? Good, Ida. They're going to attract, right? So positive and negatives are going to attract. And so what's cool about protein folding is that, let's say you have a string of 20 amino acids. You start to have these interactions between R groups, right? And so it's going to cause, like, let's say I have a positive and negative on opposite ends or near each other. They're going to be attracted to one another, right? They're going to bend down. And then the other ones who maybe are negative and negative, they're going to stay away from each other, okay? And you get this intricate folding that starts to happen. And it plays the most amazing role in making your protein the perfect protein to do its job. I wrote a couple other things too. If you're acidic and basic, you're also going to be polar, obviously, which means you're going to interact with other charged things. You're going to like water, right? So, and you're going to engage in ionic bonds with one another. So this positive and negative charge would interact. And hold on, here we go. They'd interact with one another and, oh no, where'd it go? I had a good picture of it. Maybe here it is. Did it already show up? There it is. Okay, so there's my cartoon picture of positive and negative charges are going to interact and make an ionic bond right there. And so if the environment starts to change, for example, let's say this is a protein involved in digesting your food. Okay, so it's a protein that's found in your saliva that starts to break down your food. And it has an optimal pH or an optimal temperature, let's say, right? If things change, it's going to influence that positive or negative interaction, right? So if we put it in a really acidic environment, you're going to start to change the behavior of each of those R groups, and you're not going to get the proper folding. Even one misfold will change your entire protein. And so they're pretty susceptible to environmental changes. That's why if someone has a fever, fevers are good in the beginning, but if you have a fever of like 103 or 104, your doctor will usually ask, well, how long have you had this fever? Okay, because in the beginning, a fever is good. It's your body's inflammatory response to hopefully slow down the multiplication of the virus or bacteria that's inside you. So it's not a bad thing. It's your body's response and telling you that, like, hey, I'm putting this immune response on. I'm doing something. However, there's a lot of proteins in our body, right? We've got digestive proteins, especially. We've got proteins doing literally everything, right? If you have a fever for too long, you're going to start to unwind a really intricately folded protein, which would be pretty bad, right? That's why maybe if you're starting to feel kind of sick and then you eat something, maybe food doesn't even taste very good. And some of that might be due to our proteins not folding or not acting the right way because of the environment. Here's just another picture I drew showing the protein interaction. So instead of drawing circles for my amino acids, I'm just drawing it as a ribbon now because I want us to focus on the bonds rather than the amino acids. So here are just 4 examples of what kind of bonds can start to happen when a protein goes from primary structure, that beginning long strand of amino acids. So ionic bond, like we went over. You have hydrophobic interactions as well. So any amino acid that's hydrophobic, which means it's nonpolar, right? It's not going to be charged. It's going to not want to be near charged things. Those are all going to kind of crimp in toward the middle. So the middle of your protein is generally filled with hydrophobic amino acids. And these amino acids aren't changing order. They're stuck in the order that they're in. So when it starts to fold, it becomes this really cool structure. It's a 3D protein structure, but that can be easily influenced by a change in the environment. You also have hydrogen bonding just between the hydroxyl and carboxyl groups of different amino acids. And some proteins have this really strong bond called a disulfide bridge, which can be between there's 2 amino acids called cysteine that have sulfur in it. So they form a really strong covalent bond. Okay, so we have hydrogen bonds, ionic bonds, covalent bonds, hydrophobic interactions. And any of those can be disrupted by a change in the environment or a change in your actual amino acid sequence. Okay, so if you haven't done anything on proteins yet in your class, an example of a protein would be enzymes, right? So enzymes do all the work in your body. If you've ever heard of the enzyme like catalase, which is in your blood, or enzyme pepsin in your stomach, right? It's a very acidic environment. It has a very specific shape. So this is showing it as kind of like a Pac-Man structure, right? So let's think of this as like a 3D shape with 3 little teeth in it. The idea is that specific shape fits a substrate perfectly. Okay, so if it's your job as a protein, you're an enzyme, it's your job to break something apart, right? So something comes in, you're supposed to break it up. If for some reason you've been altered, because your amino acid sequence is altered, or maybe because the environment changed, it got really hot, pH went low, pH went high, your active site's not going to look like this anymore, and you will literally not be able to do your job. And so there's a ton of diseases, cystic fibrosis being one of them that I think of off the top of my head, where it's one little protein, right? One little mutation in the DNA, and it causes catastrophic effects, okay? Which creates a huge mucus buildup in the lungs. Maybe I'll write an FRQ about cystic fibrosis because I think it'd be a really good example, and you'd have to explain it, what's the cause and effect of that change in protein. I'm gonna write that down for myself to make a FRQ for you guys because I think that'd be really helpful. All right, okay, we're almost done. We're almost to our our multiple choice questions. So nucleic acid structure function. Okay, so we know our structure. We went over that. We talked about we've got a phosphate, sugar, and nitrogenous base. Okay, the bases are forming the middle with hydrogen bonds, right? It's not a covalent bond because if it was, it'd be really hard to open up DNA to read it when we're either making protein or to break it apart and copy it and make new DNA. So, for example, when you get into the cell cycle, we'll talk all about how, like, if you get a cut, right, like a paper cut, my children always want a Band-Aid right away, and they like think paper cuts are the worst thing ever. But then literally the next day, you couldn't see where the paper cut is because the cell has regenerated, made brand new cells just like the old ones. And so each of those cells has DNA inside of it. Well, where did that DNA come from? It came from the original cell. So if that middle of like your ladder right here didn't have hydrogen bonds, it'd be really time consuming and energy consuming for a cell to rip it apart, copy it, put it back together. So the hydrogen bonds are actually there for a reason. It's perfect structure and function. The other bond we see is covalent bonds between the, let me see where my pictures are, sorry. There we go. So we have a covalent bond between our phosphate and our sugar. So if you look at this very top line here, where it says 5' end, 3' end, we've got phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So that's where you may have heard your teacher call it a phosphate sugar backbone. It's called a backbone for a reason, right? Our backs are supposed to be strong. We have this vertebral column that keeps us upright so that we're not just collapsing, okay? So backbone of DNA, think of it as something very strong. It's like covalent bonds, phosphate, sugar, right? Hydrogen bonds in the middle. Even in my diagram here, you can kind of see them. They're dotted lines to represent that they're a little weaker and that they're easy to break apart, okay? Something we'll get into in much more detail the next stream on Tuesday will be what is 5', what does 3' mean? Like what on earth is that? And so we'll talk about carbon numbering and why that matters. But basically, if we were to give you the gist of it, it's necessary for replication of DNA. It's called semi-conservative replication. So the idea is they run anti-parallel to one another. So one end has 5', the other end has 3'. So that way, when you break it apart and copy it, you have two perfect identical sets of DNA. If they weren't that way, you'd have to copy it and then flip the entire strand over, and it'd be really energy consuming. And so luckily our cells do not do that. So again, here's a precursor of what 1, 2, 3, 4, 5 numbering means. If you took AP Chem, you're like, I know this is great. You know what carbon numbering is, but we care about it in biology too. And so it's gonna make a lot more sense, I promise you. But if you understand what we mean by 5 to 3', you will be in such great shape when we talk about DNA replication and we're talking about leading and lagging strands and all that other stuff. So don't skip over this. Don't think that this isn't important. So tune in on Tuesday because we're going to really get into it. All right. Now, what happens if structure is altered? Okay. If DNA is altered, it's usually altered not within the phosphate sugar backbone because like we talked about, it's really, really strong. Okay. It's not actually being copied. It's not being read. Okay. It's the nitrogenous base that is the information for everything in your whole body, right? Your DNA is the information, okay? So if something is mistaken, so if you look here, it's identical all the way across, but down here we have an A that's been replaced with a C, okay? That's a single mutation, single nucleotide mess up, but it's catastrophic because we have a new amino acid now. Thankfully, our amino acids are very redundant. There's only 20, right? So we'll get to codon charting later, but basically some mutations can happen and nothing is affected because there's a bunch of different sets of codes that code for the same amino acids. So sometimes you get lucky and it doesn't have an effect, but sometimes it'll tell the protein to just stop and it won't make any more. And we'll get into all those different types of mutations later, but definitely if DNA is altered, protein is most likely going to be altered or could be altered. Shouldn't say that. Okay, so what if the amino acid structure is altered? You should be able to explain. You could even just choose one of these bond types, to be honest, unless the AP biology question gave you information like, oh, it went from having a hydrophobic amino acid to a hydrophilic. Explain what that's going to do, right? So if they give you information, then absolutely use it. But if it's just a hypothetical, predict what would happen if something was altered, then choose which one you know best, right? If you feel like you know the disulfide bridge really well, explain how that's going to affect the shape of the protein. If you can talk about hydrophilic things or hydrophobic or ionic bonds and give examples, then talk about that. But the idea is, okay, if the structure of protein's altered, it could be because of a change in pH. It could be because of a change in temperature, right? Temperature is going to start to break bonds, especially if the temperature stays that way. Okay, because the idea is too, this sequence of amino acids, it kind of looks like a bead on a string. That sequence was put in place by the instructions of DNA. Okay, so if you could even lead it back to that and say, well, if there was something wrong with the amino acid sequence, then that means something happened with DNA. And if you could talk about that, that'd be great as well. Okay. Can anybody name this disease? If you look at the picture, if I'm looking at, it's a red blood cell. You can name the disease. Okay, yeah, sickle cell anemia, absolutely. It's a big example of DNA mutation giving us a new amino acid. That amino acid has a different structure, and so it's going to fold differently, okay? So glutamine it's actually glutamic acid. It's polar, okay? And valine is not charged. So we're going from something that's polar to something that's not polar. And so when we have protein amino acid interactions, it's going to completely change the shape of a protein. And so that changes the shape of a red blood cell. Okay, so the red blood cell, when you get into that later too as well, it's really just proteins inside of a cell. There's no DNA in your red blood cells. There's not a whole lot of organelles. So if protein is messed up in there, it's going to mess up the shape of the actual cell as well. So here's just another picture of this R group becomes this R group, and boom, just from one little mutation, you have a disease. Okay, let's go. We have just a few minutes left, so let's go through some practice multiple choice questions. Number one, and you can just answer it A, B, C, D. So number one, proteins are composed of linked. Is it A, nucleic acid, B, phospholipid, C, amino acid? Great. Yep, composed of amino acids. That's kind of a softball question for sure, but just making sure everyone's with me. Number two, enzymes that work to help digest your food are an example of A, polysaccharide, B, nucleic acid, C, fatty acid, D, protein. Great job. Everyone's saying D. Excellent. Proteins. Very, very good. Again, that's kind of a softball question, but we're leading up to more difficult ones, I promise. Okay, hydrogen bonds occur A, between nonpolar substances, B, between adenine and thymine, and those are your DNA bases, C, between phosphate and deoxyribose in DNA, D, when a hydrogen and an oxygen and a water molecule share electrons, or E, between carbon and hydrogen and a molecule of methane. And for each of these two, the ones that are incorrect, be comfortable being able to explain what kind of bonds are made or where you see these things happening. And so it may seem like an easy question, but make sure you're able to look at each option and talk about them individually. So the answer for this one, hydrogen bonds occur between the bases, so between adenine and thymine. Okay. If you were struggling between whether or not it should be D, remember that the intramolecular bond, intra is an INTRA, hydrogen and oxygen have a covalent bond within one water molecule. Okay, so water molecules form hydrogen bonds with other water molecules, which is your intermolecular bonding that's between molecules. Okay. Here's a good review one. Which of the following statements is or are true with regard to a polymer of 6 glucose molecules? So it could be more than one answer. So glucose has the chemical formula C6H12O6. So if I have 6 glucose molecules that are made into a polymer, what's my chemical formula? I gave this as a test, a similar question to this as a test question to my AP students every year, and they always get it wrong because they speed through it and they just kind of assume they're good to go. Okay. So let's think back. If we're joining, all right, most people are getting it. If we're joining two glucose molecules together, right, we have two molecules, but then one bond, okay? We've had to take a water out of that, right? We have dehydration synthesis to make that one bond. So if I have 6 molecules, and this is a good example of where drawing something out might help, like draw 6 circles to represent glucose and make a line across each one to say, okay, well, I'm actually only doing 5 bonds. So I have 5 moments of dehydration synthesis. So 5 water molecules were made. Okay, so think about, well, what does 5H2O look like? Okay, so what happens is we can't just multiply our subscripts here and say, oh, it's times 6, right? What happens is we've lost 10 molecules of hydrogen, or atoms of hydrogen, and 5 of oxygen. Okay, so 5 water molecules were taken out. So we can't just multiply. So chemical formula is that for 2. And then for the monomers, we're joined via dehydration synthesis. So this is kind of asking a couple things all at once. Okay, just be careful, read things carefully. Okay, last multiple choice question. 20 common amino acids differ from each other in the composition of a covalently bonded side chain known as A R group, B dipeptide, C polypeptide, D amino group, and E carboxyl group. So what do each amino acid differ from each other with? Oh, you guys are fast. Awesome. A, R group. Good. The R group is that unique one. Very, very good. Okay, we are running out of time. I'm supposed to end at about 50 minutes. So here's your FRQ prompt. You take a screenshot of it or maybe take a picture of it. And so what I'll do, because we're running out of time, I will add this to my Tuesday stream. I think I'm the next streamer anyway. So we'll start off our nucleic acid stream going over how to answer this. So ideally, try to do this on your own. The FRQ says, physical form of cells and organisms is influenced by structural polymers. Part A, for each below, describe the structure and role of each in an organism. And B, predict the cause and or effect of a disruption in one of these polymers. Okay, so there's 2 parts. And for each part, it's wanting you to look at these 2 examples, DNA and cellulose. So take each part at a time, describe the structure and role of each. More detail, the better. And predict the cause and or effect of a disruption in one of these polymers. This is a short FRQ. Go ahead, take a look at it.